Now let's take a few minutes and talk about the individual organelles that we find in eukaryotic cells, including human cells. Remember, organelles are either double membraned or single membraned. The nucleus and the mitochondria and the chloroplast are examples of organelles that have two membranes around them. Now we refer to the nuclear membranes as an envelope because just like a paper envelope, those two membranes come together and connect with each other everywhere that there is a pore in the nuclear envelope. The mitochondria has inner and outer membranes, and the chloroplast too has inner and outer membranes. I have the chloroplast in parentheses here because we're not going to talk about the chloroplast in any detail. And that's because human beings don't have chloroplasts. Remember, chloroplasts are the organelle that performs photosynthesis in organisms that are capable of that. Now, the single membrane organelles include the endoplasmic reticulum, and there are rough and smooth varieties of that, the Golgi apparatus, the lysosomes, the peroxisomes, and what are called transport vesicles. In previous courses, you may have learned a little bit about the function of these organelles. Rough endoplasmic reticulum is involved in the production and the modification of protein. Smooth endoplasmic reticulum is involved in lipid synthesis, calcium regulation, and also the detoxification of several different compounds. The Golgi apparatus is involved in the synthesis of complex carbohydrates, and it also plays an important role in the modification of certain types of protein with carbohydrate. Lysosomes are the digester organelle in the cell. Lysosomes break down and recycle old materials that the cell no longer needs. Lysosomes are also capable of destroying any pathogenic organisms like bacteria that might gain entry into the cell. Peroxisomes are a lot like lysosomes in that they're also going to be involved in degrading material, but peroxisomes are more specifically involved with things like the detoxification of hydrogen peroxide. Hydrogen peroxide is a toxic material to living cells, and the peroxisome is able to degrade it and detoxify it. Peroxisomes are also capable of breaking down some molecules that lysosomes are unable to deal with. For example, certain alcohols will be degraded by peroxisomes. There are certain drugs that are broken down with peroxisomes. And finally, there are some unusual molecular forms of amino acids that the cell can't use that a peroxisome will break down. And finally, there are the transport vesicles, which, like the name suggests, are involved in transporting molecules throughout the cell. We'll take a few minutes and we'll talk a little more about the structure and the function of some of these organelles. We'll start with the nucleus. The nucleus, remember, houses the DNA inside of a cell. Now, all cells have DNA, but only eukaryotic cells have a nucleus. The nucleus offers protection to this critically important molecule. Now, DNA doesn't simply float in the nucleus as a double helix, however. Instead, it's carefully folded up around a type of protein called histone proteins. Once it's all folded around these protein molecules, the DNA is carefully stacked into a very compacted structure called chromatin. At other times during the life of the cell, the DNA is folded up even more and creates what's referred to as a chromosome. The DNA molecule, you could argue, is the single most important molecule in a cell. The DNA molecule performs two very different but very important processes for the cell, and those processes occur in the nucleus, where the DNA is housed. For example, DNA is used for the process called replication. This process occurs whenever a cell is preparing to divide and create two new cells. Each one of those two new cells is going to need its own copy of the DNA molecule. So replication is the process where the DNA molecule will be replicated or copied. DNA, in other words, gets used to make more DNA during replication. The other process that the DNA is used for is part of what we call gene expression. The process called transcription uses the DNA molecule to make a molecule called RNA. Transcription, like replication, occurs in the nucleus. Now, when you look into a nucleus like the one drawn at the bottom of this slide, most of the time in the life of a cell, the DNA is just going to look very homogeneous. It's going to look very uniform, just sort of spread out, occupying the space inside of the nucleus. This is chromatin. This is the folded up and stacked version of DNA. Sometimes you'll notice a darker area inside of the nucleus. This is a particular region of the DNA, of the chromatin, called a nucleolus. Now, depending upon the point of the life cycle for that cell, when you examine it under the microscope, you may or may not see a nucleolus inside the nucleus. You have to catch the cell at a very particular time, in other words, to be able to see the nucleolus. Now, the nucleolus forms because a process is occurring inside this particular region of the chromatin. And that process is the production of a type of RNA called rRNA, or ribosomal RNA. Remember, ribosomal RNA is going to become part of the ribosome. And the ribosome, of course, is going to be used to build the proteins for the cell. Sometimes you will see one nucleolus inside of a nucleus. Sometimes you'll see several of them. The plural form of the word nucleolus is nucleoli. In this image, you can also see on the surface of this nucleus all these little holes. Those are called nuclear pores, and those are the structures that carefully regulate what can come into the nucleus and what can leave the nucleus. Remember, one of the most important, if not the most important molecule in the cell is housed in this structure, the DNA. So it's in the best interest of the cell to control what can come in and what can leave. It's in the best interest of the cell to protect the DNA from anything that could harm it. These holes, these pores that you see on the outside of the nucleus, these are not just tunnels into the nucleus. They are gated, they are protected, and they have to be opened up, particularly for individual molecules to be allowed to enter or exit. You can just make out in this diagram that there are two layers to the membrane surrounding the nucleus. There's an outer layer and an inner layer. We refer to this double layer as an envelope. And again, the reason we call it an envelope is because the two layers come together and join each other everywhere that there is a nuclear pore, just like a paper envelope. On this slide, in the upper left-hand corner, you can see a smear of blood, human blood. We're looking under a light microscope here. This would be a bright field view because the cells are darker than the background. There are a couple of white blood cells visible. One is right here and one is right here. But most of these cells are red blood cells, or RBCs. The technical term for a red blood cell is an erythrocyte. These are the cells that travel in your bloodstream and carry oxygen to your tissues and then remove carbon dioxide from your tissues. Red blood cells are a really interesting type of cell because they mature and they lose their nucleus. If you were to examine one of these individual red blood cells under a high-powered electron microscope, there would be no nucleus in that cell. The reason that they lose their nuclei is because they are highly, highly specialized in the job that they're going to do as they circulate in the bloodstream, and they simply don't need their nucleus anymore. The cell rids itself of the nucleus because it doesn't want to spend any time or energy maintaining the nucleus. It doesn't need it anymore. And that tells you something. That tells you that a circulating red blood cell is not going to be doing either of the two things that happen in the nucleus. It's not going to be doing DNA replication, and it's not going to be doing transcription. Everything these cells need, they have. They don't need to make any additional proteins. They circulate for about 120 days in a human being, and then they're degraded and replaced with new cells. Now compare that to these white blood cells. The dark staining area inside these two cells is the nucleus. White blood cells don't lose their nucleus as they mature, and that's because they still need their nucleus. White blood cells can divide, and they need to express their genes. They need to make proteins throughout their life cycle. Remember, white blood cells are a critical part of the immune system, and so they need to be able to change during their life cycle, unlike the red blood cell. Now, on the bottom of this slide over here, you're looking at some more of these epithelial cells. These cells, too, were taken from the inside of someone's cheek. These cells look a little bit different, though, from the ones we saw earlier. These cells have lost their nuclei. Epithelial cells are present in layers upon layers. If you think about your skin, for example, and we'll be talking about the skin in detail in a couple of weeks, but you have several layers of epithelial cells in your skin, and the outermost layer, the layer that sort of flakes off every day, that layer is made up of these types of epithelial cells. These are called squamous epithelial cells, and they have undergone a process where they lose their nuclei. At this point in their life, they're very flat. They have very odd shapes. I think you can see that on this slide. And they are no longer dividing or expressing their genes. So they no longer need their nucleus. We'll leave the nucleus now and we'll start talking about the mitochondria. Remember, the mitochondria are the place in the cell where ATP is being made. If you remember, ATP is made through a series of complex metabolic reactions that we refer to as cellular respiration. Human beings do a form of cellular respiration called aerobic cellular respiration. We do it in the presence of oxygen, in other words. This long series of complex reactions is what produces this critical molecule called ATP. Now you should know the name of ATP, not just the abbreviation. Remember, it stands for adenosine triphosphate. When you look at a mitochondria, you're looking at another one of these double-membraned organelles. The mitochondria has what are referred to as an outer membrane and an inner membrane. And you can see both of those in this image. Now, if you were to look at the inner membrane all by itself and the outer membrane side by side, you would see that the inner membrane has a much greater surface area than the outer membrane. It's almost like if you were holding a baseball in one hand and a soccer ball in the other hand. The soccer ball, of course, has a much greater surface area than the baseball does. If you were to take the surface of the soccer ball and try to insert it into the baseball, you'd have to fold it up quite a bit. And that's what's happening with the inner mitochondrial membrane. It's folded up quite a bit in here in order to fit, essentially. Now, anytime you see this sort of extensive folding of membrane, it's there for a reason. It's providing surface area for some type of function. And when it comes to the inner mitochondrial membrane, it's providing surface area to allow for lots and lots of aerobic cellular respiration to occur. All of the molecules that are involved in that process are found in this membrane and in the cavity that it surrounds, called the matrix. So the inner membrane is larger than the outer membrane. It has a greater surface area. And the folds that are created by that inner membrane get a name. We refer to those as cristae. When you put one membrane inside of another membrane, you end up creating multiple compartments in here. So we give those a name as well. For example, the space between the outer and the inner mitochondrial membrane is called the intermembrane space. That's a nice name, makes sense. That term inter means between. So between the membranes. This space that lies within the inner mitochondrial membrane, this is the matrix. Now, inside the matrix, there is a gel-like fluid, a lot like ordinary cytoplasm. And this gel-like material contains a couple of other critical structures. It contains ribosomes, and it contains small circular pieces of DNA. That's right, mitochondria have their own DNA. We refer to mitochondrial DNA as mtDNA. Mitochondrial DNA is a fascinating little piece of DNA because it's passed down from mother to offspring directly. There's no influence of the father in there at all. We use this fact, this maternal inheritance, in order to do some forensic science. We can often trace back DNA samples based upon what's in the mitochondrial DNA instead of the nuclear DNA. Now, you might ask yourself where DNA comes from in a mitochondria. Why would a mitochondria have its own DNA molecule? And the answer is that we believe that mitochondria were actually once a free-living organism. We believe that there was once a cell surrounded by the inner membrane that had cytoplasm and ribosomes and DNA in it. And through some evolutionary process, it was consumed by a larger cell. And in that consumption process, in that eating process, that little cell got wrapped in a second piece of membrane and created what are now the mitochondria. This is referred to as the endosymbiosis theory, that mitochondria were once free-living organisms and that they were engulfed by a larger cell and became part of that larger cell. Endosymbiosis means the symbiosis within, and it refers to the fact that both organisms benefited when that little cell was eaten by that bigger cell. The little cell got protection from the bigger cell, and the bigger cell got the bonus of what the little cell was making, which was ATP. On this slide, we're looking at a couple of EM images. This image on the upper left-hand side of the slide shows us a mitochondrion. Mitochondrion is the singular form, and mitochondria is the plural form. Most cells have multiple mitochondria in them, but it depends upon how much energy that cell needs every day. There are some cells that have much higher energy needs than other cells. Cells like skeletal muscle, again, those cells have a very high requirement for ATP. They burn through a lot of ATP every day. In comparison, something like a fat cell, which has a much lower metabolic rate, would have many fewer mitochondria in it. In this image, we're looking inside of a cell and we're seeing this sort of potato-shaped structure. This is the mitochondria. Now, it doesn't look quite like that perfect drawn image that we saw a moment ago. Actual cellular images rarely look like the perfect way that we draw them. But you can see that this structure has a membrane all the way around it, and then it has these strange folds inside of it. These are the cristae, these dark, sort of finger-like folds that you can see. The light-colored material here and here, that's the matrix. The dark circles, those are ribosomes. And the larger structures are most likely the mitochondrial DNA, the larger black structures. Now, mitochondria are almost shaped like kidney beans. So depending on how the cell gets placed on a slide to be viewed under the microscope, depending on how it got placed, you may view mitochondria elongated like this, or they may look more circular if they are viewed end on end. In other words, sometimes you're going to view the longitudinal section of a mitochondria, and other times you're going to view a transverse section of mitochondria. It all depends on how the cell sort of landed on the slide. Now take a look at this image down here. This looks sort of like another image that we looked at earlier, but this one is colorized. So they have added artificial color to this section of skeletal muscle. Again, you can see the individual filaments in these red structures all lined up on top of each other, creating these sarcomeres. I want to draw your attention to the space between these filaments, the space that is colorized to this brown and green. These structures right here, in between the sarcomeres, these are mitochondria. You can see that, again, they've been colorized, and it's difficult to see much detail in here. But we would expect to find lots and lots of mitochondria inside of a muscle cell. So this process that we call cellular respiration is going on in this organelle, and it's producing this very important energy currency molecule that we call ATP. Now there's just a couple of things I want to mention here about cellular respiration. Cellular respiration involves a carbohydrate called glucose. That glucose molecule is going to be broken apart inside the mitochondria. All of the covalent bonds that are in that 6-carbon glucose molecule, all of those molecular covalent bonds are going to be broken apart. And the electrons that sit in those bonds are going to be released. So the glucose molecule will be taken apart bond by bond by bond until the only thing that's left is carbon dioxide. We refer to that long process as oxidation. We call that the oxidation of glucose. Now, all of the electrons that are set free from that glucose molecule, they're going to be used to do something else, and that something else is build ATP. So the two products that are produced in aerobic cellular respiration are carbon dioxide, which is a waste gas for human beings, and ATP.